Yes.
Wait,
So
why are we talking about bioluminescence?
read the chat, Joshua. Come on.
I can't see the chat. Here, I'm here.
So let me.
Oh,
No. Where is it? So this is actually
I'll turn
it's
and
a
fix
new
it.
technique within neuroscience called bioluminescent optogenetics.
Oh, it's the ambient light optogenetics,
You've heard of this?
basically. Yeah, I've heard of it because you told me about it.
Oh, okay. So yeah,
I have
deking. Basically, it's the same thing that you would use in other bioluminescent products. Let's take a step back. So the first thing you have to consider is we have optogenetics. Like we can activate neurons in the brain with light. The trouble is, how do you get light into the brain?
Wait. We have to tell also that we can
No,
activate
it's fine.
neurons.
It's fine.
It's fine? It doesn't matter.
Don't get
that we
into
are
boring
connected.
details.
Okay.
So you have optogenetics. You can put stuff in brain cells, and then when you shine light on them,
Yeah.
brain cells become active. Yay!
Yes.
Could do that. Put them in your midbrain, and whenever you shine light, you become happy. Super cool.
Yes.
We're not really testing this on humans yet.
We only have
But yeah, so the other
electricity.
thing is, like you said, we have made plants and fish and mice that glow in the dark, or in a black light. We have bioluminescence. So there's this really, I don't know, some researchers decided that, we can combine both of these things. And that's what I've done. So they have the luminescence you would find in, glow in the dark food products, let me call it. They put that in the same construct as the optogenetic thing. Let me find a photo for it in a second. And it make more sense. That looks something like this. And basically, you give the stuff that makes the bioluminescent work. You give the luciferin substrate. That will activate the bioluminescence. So your neurons will start to glow, and then because they start to glow, they will activate their ion channels, and they become active or silenced. So it's a way of combining optogenetics and bioluminescence. It's marvelous. In this case, they don't actually have to shine a black light on it. It's I'm not an expert on bioluminescence. It is some substrate of luciferase. I don't know. If you know Linus Tech Tips, they did just a whole video on bioluminescence in computers. Tissue cultivation, all sort of.
Cool.
There's all sorts of applications for bioluminescence now. One of my favorite ones, actually, let me try to find that.
But this one is tied to a ion channel.
Yes. So this one is tied to an ion channel. Wait, let me share this. So this is a video from Linus Tech Tips where they were making a bioluminescent coolant for using computers. Which is mind-blowing. Let me share the link for that. But anyway.
That's cool. Wait. Does that change like color when it's warm or cold or something? Or?
No.
Oh,
You activate it, and it's basically an alternative to glow sticks.
Yeah,
that's the idea.
Yeah. Okay.
But yeah. So what they can do with this is activate neurons whenever the compound, I forgot what it's called, CTZ. Whenever you apply that, your neurons become active.
So it's like a combination of optogenetics and chemogenetics, almost.
So that's why I put it in the title. Yeah, exactly, Dickie. So I was surprised to see that at just the right time. So DREADS has been an alternative way to activate neurons. Basically, with DREADS, you apply a chemical that only binds to a very designed receptor. That's where the name comes from. Designer receptors exclusively activated by designer drugs. So you have a very special drug that does nothing, but you put it in and you give a very special receptor in your neurons, and those are only activated. So you can very selectively activate certain cells. This is kind of the same thing, except you apply the compound and the neurons become active. It's you apply the compound, the neurons start to glow, and then the glow activates the neurons. It's sparkly dreads.
Sparkling dreads.
Sparkling dreads. It's not real dreads. Could be used in Parkinson's. Absolutely, D. King. So again, this has not been applied to humans yet, as far as I know. One of the hallmarks of Parkinson's disease is that we lose dopaminergic neurons in the midbrain, of course. The mechanisms underlying that are still up for debate. It's thought to be oxidative stress, or mitochondrial stress, but I don't think we've really nailed it down yet. But one of the things you could theoretically do is stimulate the remaining neurons with bioluminescent optogenetics and keep them active, or better yet, go to the downstream targets that are no longer activated enough. You can give an injection, and this stuff will work for about an hour. I don't know if they can prolong that.
But even better, this is like an engineered thing. So you can eat pills with the compound that crosses the blood-brain barrier. Like you don't have to.
That's exactly what we're doing. So there is a compound that crosses the blood-brain barrier, and if you have a pill that gives that off slowly enough,
so it will be very easy.
Yeah, because right now the treatment for Parkinson's disease, the most common treatment is levodopa, L-Dopa, which is basically a sort of precursor to dopamine, because you don't have enough dopamine in your brain in Parkinson's disease. It's a precursor to dopamine, and that gets converted into dopamine in your brain, and then does the things that dopamine does. But alternatively, if you can have this stuff in your cells activated, that might work just as well.
It might be, if it ever gets approved for humans, it might be more localized than the levodopa or other treatments, because you can apply this only in the midbrain.
Exactly. So the alternative treatment for Parkinson's disease, what used to be, was anticholinergic drugs. So because you have this balance of acetylcholine and the dopamine would go down because the dopamine neurons die, and then you had too much acetylcholine. So people would apply muscarinic acetylcholine blockers. So your acetylcholine would go down. The trouble is that was never localized enough. So, and you kind of use acetylcholine everywhere. So, you lose muscle function. It has all sorts of horrible side effects. So we don't do that anymore. But if you could put it in just the right cells, and this is one way to do that, and it could be used for a host of different applications, ultimately. Alzheimer's disease might be treated.
And the depression thing, where they also use the deep brain stimulation. I don't know exactly in which areas they do that. But
I'm not sure where depression is localized. But
no, I thought they also did. I thought they also stimulated something in the midbrain.
Possibly. I thought subthalamic nucleus, but I might be wrong there. But I think for Alzheimer's disease, which is more about forgetting or general cognitive decline, it is usually the neurons in your cortex, slightly different from what you would use for treating Parkinson's. But it's all sort of very cool stuff that we may be able to do in sometime in the future. Forgetting things, that is that's a difficult one, and I'm not sure how far along we are to really, because we don't really understand how memory works to begin with.
And we need to forget some things, right? And our body gets older, so our cells cannot keep functioning at an optimum. So I don't think we can fix everything there.
Well, there is a lot of work being done on that, but all right. Thanks for your input, Dicking. It's a good question. Shall we go on? Next topic.